First, we are going to study about our microsporogenesis. The term says that microsporogenesis is consist of one term is your microspore, otherwise known as your pollen grain, and your genesis means your formation. And ultimately, it shows that it is a formation of pollen grain. We just know the mechanism first, and I tell you one thing that not to be panic about the term and these types of diagrams. It is quite easy to know and easy to understand also. First we saw the diagram. Nextly we are going to about the mechanism. Going to study about the mechanism. It is your PS structure of your anther. In your previous structures, you already know that the anther is consist of a bilobe structure and a filament. And the bilobe structure there is a transverse section which is given in your diagram, and this is the outer walls, just the walls of the PS structure, and it is consist of a filamental structure which is your connective structure and 4 lobes, and it is bilobe structure and 4 pollen sacs actually, and these are the pollen sacs, and these 2 lobes, it is single lobe and it is another lobe. I just draw a enlarged view of the wall structure here, and the enlarged view shows that there are 4 types of walls actually present here. One is your epidermis, another one is your endothecium layer. These are 2 consecutive lobes, and after epidermis, it is the first outer wall of your microsporogenesis. Formation of a microsporogenesis, it is the first outer wall epidermis layer, and next one is your endothecium layer, and then a number of middle layers are present which are formed from the endothecium. In the mechanism part, I also clear you about your middle layer, and next one is your tapetum, and last one is your formation of microspore motor. We are going serially about your first structure, then second one, next your third, then your fourth. First is your epidermis layer. It is the outer layer of your structure of your anther, and I also told you that it is the outer layer. It is a linear structure which are arranged consecutively. The number of cells are arranged consecutively, and this epidermis layer is consist of mass of undifferentiated cells which are homogeneous in meristematic cells, homogeneous meristematic cells, and these cells are otherizedly first, because these cells are matured condition, there is no cell division that takes place, and the cells are quite equal to look like, and the cells are quite equal to look like. It means every cell are equal from the view. These are equal cells. So after some time, the equally mass of cells are differentiated to archisporial cells. Archisporial formation takes place. Archisporial means this is the prominent the formation of your prominent nucleus and dense cytoplasm. Means the cells are prominently differentiated and able to differentiate another cell and another structure also. The cells are able because there is a prominent nucleus takes place and dense cytoplasm also placed. So and the next step, there is a division of your pericyclic division takes place. Pericyclic division means the division in which cells are planically divided. Pericyclically divided means say this is your cell and this is your nucleus part. If you divide like that, it is your pericyclic division. If 2 cells are there and a pericyclic division takes place, such like that, if 3 cells are formed, here nucleus is divided, cytoplasm is divided, and formation of your 2 cells takes place. But in a single layer, if we say that this is your epidermis layer and this is your endothecium layer, in this structure, in epidermis layer is formation of epidermis layer already takes place, and in your endothecium there is the formation of your archisporial cells takes place. Archisporial cells means the cells in which the pericyclic division takes place. Pericyclic division means there is a simple division takes place in these cells. That means there is a multicellular cell, there is the formation of multicells that takes place. That means there are 2 division of cells. Each cells are divided in pericyclically, and its nucleus, prominent nucleus and cytoplasm components are also divided. And it is the formation of your archisporial cells takes place. Then in the archisporial cells form 2 types of cells. One is your primary parietal cells, otherwise known as your PPC. Another one is your PSC, otherwise known as your primary sporogenous cells. cells. Two types of cells are placed in, one is your outer part, another one is your inner part. Below epidermis, below the epidermis, primary parietal cells takes place. This is your epidermal layer, and in this layer, these parts are fulfilled by your primary parietal cells, and your second part is which is your inner side part. If you say one example, if you take, this is your cell, this is your outer part, this is your inner part. If the cell division takes place and cell deposition takes place, deposition are in bilobed structure, as it is a bilobed structure division. The cells are divided periclinially and deposited two sides. One is your outer side, one is your inner side. The cells which are deposited in outer side, I repeat that. The cells which are deposited outer side is known as your primary parietal cells. The cells which are deposited inner side, means towards the cells, is otherwise known as your primary sporogenous cells. I also written in here, your primary parietal cells are deposited in below epidermis, and primary sporogenous cells are deposited towards the center. That means towards the center region. And nextly, divisions are takes place. Both sides are divided takes place, and obviously, as it is your epidermal layer below the epidermis layer, it is obviously a formation of next steps are takes place. And next steps are your periclinial division and repeated anticlinial division takes place in these cells, and formation of your endothelium, middle layer, and tapetum takes place. Here, tapetum is very sensitive layer, and a lot of works are also there. In the mechanism part, I am going to tell you about your tapetum. To say that, I also clear one thing that the repeated divisions are takes place in these cells. The two types of cells are repeatedly divided to form your sporogenous tissues, and here the deposition of the cells takes place. And for the deposition, there is maximum 2 to 3 layers are formed in a single cellular structure. And first, there is the division of your epidermis takes place. Next, your endothelium takes place, and the 2 to 3 layer are forms your middle layers. Middle layers is nothing but the archisporial cells which are divided to form your PPC and PSC. The two cells are divided outer side and inner side, and formation of your layering is takes place here, and it is otherwise known as middle layer. Nextly, next to the middle layer, there is the formation of your tapetum. It is the mostly outer layer of your microspore mother cell, because after crossing the tapetum, you reach to the microspore mother cell, which is self-dividing cells, your microspore mother cells, which is the formation of your meiotic divisions obviously. And when the formation of your primary sporogenous takes place towards the center, next your sporogenous cells formation takes place, and next one is your tapetum layer. It is just the layer which gives the nourishment to the microspore model cells or MMC cells, and MMC cells are getting divided. Here I tell you that meiotic divisions takes place. Why I am telling about your meiotic division? How I not your mitotic division? Because here in the meiotic division, microspore mother cells are already formed your 2N structure. Here we have to consider to the pollen grains, as we are studying about single pollen grains. It is N number of structure, and we also know about your cell division structure. In meiotic divisions, these types of structures are formed, and here we discussed a number of things about your mechanism. It is a simple process, consists of your 4 layers. One is your epidermis, another one is endothelium, next one is your middle layer. Middle layer is important because below the epidermis there is a number of archisporial cells or formations that takes place. Archisporial cells means it is a simple structure in which a dense cytoplasm and prominent nucleus takes place. That means the total cell is able to divide, and the divisions takes place in 2 types of manner. One is your primary parietal cells, one is your primary sporogenous cells. These types of divisions takes place in layering manner, which is otherwise known as your periclinial division, and in repeated divisions it is also takes place in your periclinial and anticlinial both. But in this case, below the epidermis, the periclinial division takes place, and a layer of cells are formed because the periclinial division formed in your outer part and inner part. Outer part, the cells which are divided in your outer part is otherwise known as your primary parietal cells, and Inner part which are divided is your otherwise known as your primary sporogenous steps. The both are divided and form a layering like structure, and before after that there is a layer formation, nourishing layer formation takes place, which is otherwise known as your tapetum, which helps the microspore mother cell to nourish and development of the pollen. And nextly the tapetum layer, and next one is your microspore mother layer, microspore mother cell formation. It is all about your microsporogenesis. Nextly we are going to study about structure of pollen. Thank you.